things about nucleic acids. DNA and RNA, the other two types of nucleic acids, where the genetic information is encoded in the molecular structure of nucleic acids. When we clearly talked about the genetic information, like the hereditary information is passed on from a cell to its daughter cell at cell division, and from one generation of an organism to the next generation through the organism's reproductive cells. So living organisms have a common feature, a common important feature, that is all living organisms use similar genetic systems. A complete set of genetic instructions for an organism is its genome, and all genomes are encoded in nucleic acids, either DNA or RNA. So these nucleic acids are the main information carrying molecules of the cell and also determine the inherited characteristics of every living thing. So these nucleic acids store genetic information and enable protein production. These nucleic acids composed of long strands of nucleotides, which consists of a nitrogenous base, a 5-carbon sugar, and a phosphate group. So coming to the nitrogenous bases and also nucleotide and the phosphate group sugar molecule structures. So coming to this slide, here the nucleic acids are made of elementary building blocks called nucleotides. That each nucleotide has three components: a sugar molecule, a phosphate molecule which has acidic chemical properties. The phosphate molecule is a acidic chemical, is having acidic chemical properties, and also a nitrogen containing molecule which has slightly basic chemical properties. So if we observe here that a sugar molecule is there, a phosphate molecule is there, a phosphate molecule which is having acidic chemical property, and a nitrogen containing molecule which is having slightly basic chemical property. So nucleotide having sugar, phosphate molecule, and a nitrogenous base, whereas nucleoside is a molecule which is containing only pentose sugar linked to a nitrogen base. So these nitrogenous bases are also called as glyoxyl amines. So a nucleoside can also be defined as a nucleotide without a phosphate group attached to it. So in ribonucleic acid or RNA, the constituent sugar is ribose. In deoxyribose nucleic acid, it is deoxyribose. So within RNA or DNA, the one nucleotide is distinguished from another by its nitrogen containing base. So in DNA, the four kinds of bases are adenine, guanine, cytosine, and thymine, whereas in uracil is only present in RNA. Along with adenine, guanine, cytosine, uracil is also present in RNA. The difference between DNA and RNA with respect to nitrogenous bases are presence of uracil in RNA and absence of uracil in DNA. Thus, in both DNA and RNA, there are four kinds of nucleotides, and three of them are shared by both types of nucleic acid molecules. Let us go to the next slide. Here, two types of nitrogenous bases. Those are classified into pyrimidines and purines. So pyrimidines are 6-membered single ring compounds, whereas purines are 9-membered double ring compounds. Uracil, cytosine, and thymine falls under pyrimidine category, and adenine and guanine falls under purine category. So deoxyribose is linked at position 3 of pyrimidine, whereas the same deoxyribose is linked at 9th position of the purine. Okay. And now the structure of DNA and RNA, which is very, very important here. Okay. In this slide, we are going to know about the structure of DNA and RNA. So if you observe here clearly that DNA is double stranded in nature and RNA is single stranded in nature, whereas DNA having two polynucleotide strands, whereas RNA is having single polynucleotide strands. So here the two strands of DNA are opposite to each other, I mean complementary strands. One is of 5 prime to 3 prime direction, and the other one is 3 prime to 5 prime direction. I mean two strands are opposite in nature. And also adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine in 3 hydrogen bonds. There is no chance of placing uracil in DNA, and in case of RNA, uracil is placed. So these hydrogen bonds are weaker bonds, and also the DNA has deoxyribose sugar. And also if you observe here in this structure, the phosphate group is there, nitrogen base along with a hydrogen bonding between two nitrogen bases. I mean two hydrogen bonds placed between adenosine and thymine, and 3 hydrogen bonds are placed between guanine and cytosine. The polynucleotide strands DNA is made up of many nucleotides connected by covalent bonds, I mean which join the 5 prime phosphate group of one nucleotide to the 3 prime carbon atom of the next nucleotide. These bonds called phosphodiester linkages, which are strong covalent bonds, and the negative charges of the phosphate groups are frequently neutralized by the association of positive charges of proteins and other molecules. An important characteristic of polynucleotide strand is its direction, I mean that polarity. At one end of the strand, a free phosphate group is attached to the 5 prime carbon atom of the sugar in the nucleotide. See, here a consequence of DNA structure and base pairing requirements is that each strand of DNA molecule contains a sequence of nucleotides that is exactly complementary to the nucleotide sequence of its partner strand. Okay. This is about the structure of DNA and RNA. It is clearly known that DNA is double stranded and RNA is a single stranded molecule. Okay. Both DNA and RNA consist of polynucleotide strands. Okay. Next. Yes. The salient features of double stranded helical structure of DNA we are going to discuss now. Okay. The salient features means, I mean the majorly the DNA molecule consists of 2 polynucleotide chains wound around each other in a right hand double helix. The 2 strands of DNA are oriented anti-parallel to each other, that is the 5 prime end of one strand is located with the 3 prime end of the other strand at the same end of a DNA molecule. And also each polydeoxyribonucleic acid strand is composed of many deoxyribonucleotides joined together by phosphodiester linkages between their sugar and phosphate residues, and the sugar phosphate backbones are on the outsides of the double helix with the nitrogen bases oriented towards the central axis. Okay. Here the diameter of this helix is 220 angstroms while its pitch is 34 angstroms. In each DNA strand, the bases occur at a regular interval of 3.4 angstroms, so that about 10 base pairs are present or arranged in one pitch of a DNA double helix. Okay. The bases are connected by hydrogen bonds. Although the hydrogen bonds are weaker, the fact that so many of them occur along the length of the DNA double helix provides a high degree of stability and rigidity of the molecule. Okay. See that diameter in the diagram, it is clearly shown that the diameter of a helix is 2 nanometers while its pitch is of about 3.4 nanometers, and from one nitrogen base to another nitrogen base it is 0.34 nanometers. Okay. The helix has 2 external grooves, one deep wide one and that is called as major groove, and one shallow narrow one called minor groove. Okay. While talking about the salient features of this double stranded helical structure of DNA, we need to talk about denaturation and renaturation. The hydrogen bonds between the DNA strands break on heating the DNA to high temperatures, nearly 100 degrees centigrade. The process of separation of DNA strands is known as denaturation, whereas renaturation is nothing but reunion of separated or denaturated strands on cooling is called as renaturation. It is called as annealing. The optimum temperature for renaturation is 20 to 25 degrees centigrade. Okay. For denaturation, we need to break the bonds. We need to divide the strands by breaking the hydrogen bonds, by heating the DNA to high temperature at 100 degrees centigrade, and also for renaturation we need to maintain the temperature of 20 to 25 degrees centigrade, and this renaturation is also called as annealing. So these are the salient features of double stranded helical structure of DNA. Next, coming to the structures of RNA. Although RNA is usually a single stranded in nature, short complementary regions within a nucleotide strand can form and pair secondary structures. So if we observe here in the second figure, see here a RNA molecule folds to the folding was happening. I mean it is linear in nature. The primary structure is linear in nature. When a RNA molecule folds to form a structure, the structure looks like here. I mean that secondary structure is determined by the base sequence of the nucleotide strand. So different RNA molecules can assume different structures because their structure determines their function. RNA molecule have the potential for tremendous variation in function. Okay, these are the structures of RNA, the primary structure and secondary structure of RNA. Well, we know about RNA as a single stranded molecule. Here in this single strand molecule, the 5' 3' strand is there with phosphodiester bonds, polynucleotide strand which is having nitrogenous base. Uracil is also present here. RNA contains uracil in place of thymine, where RNA has a hydroxyl group on the second carbon atom of its sugar component, whereas DNA has a hydrogen atom. Okay, I mean that it implies RNA is more reactive than DNA. Okay, I mean this second this hydroxyl group on second carbon atom of a sugar component makes more reactive RNA more reactive than DNA. Okay, next one, functions of DNA. What are the major functions of DNA? Okay, let us see here. Very important thing is that deoxyribonucleic acid, nothing but DNA, serves as a blueprint for life. Its primary function its primary functions are like storing genetic informations, and also for protein synthesis, and for replication, and for regulation of gene. Okay, I mean that DNA contains the instructions needed for an organism's development, functioning and reproduction. I mean these instructions are encoded in the sequence of nucleotides within the DNA molecule, and also protein synthesis. Protein synthesis, proteins are essential molecules that perform a wide range of functions within the cells, including enzymatic activities and signaling. So DNA molecule satisfies the requirement of genetic material in many ways, like by storing genetic information, by protein synthesis, by replication and by regulation. So the essential functions of DNA are storage and transmission of genetic information, and also the expression of this information in the form of synthesis of cellular proteins. So here in the diagram it is clearly representing that how DNA is going to change, okay, the pathway. So here in this diagram the pathway from DNA to protein, it is showing the pathway from DNA to protein, the flow of genetic information from DNA to RNA through transcription, and from RNA to protein through translation, which occurs in a living cell. I mean it is completely a we can call it as central dogma that we will discuss in the next slide. Okay, these are the functions of DNA. I want to recall one more time about the functions of DNA. It can replicate itself accurately during cell growth and division, and is sufficient stable so that heritable charges like mutations can occur only very rarely. It has a potential to carry all kinds of necessary biological informations, and also transmits all the biological information to the daughter cells, and the other functions of DNA. And the types of DNA, basically 4 types of DNA are there. Here I am representing, in this diagrammatic representation I am showing here only 3 forms: A, B, Z form. One more form is Z, C form is there. A, B, C and Z forms of DNA. As we all know that B is the important form of DNA that is commonly and naturally found in most living systems, whereas A form is also a right-handed helix DNA, but it has 11 base pairs per turn. We having in this A form of DNA 11 base pairs are there, and in C form 9.3 base pairs per turn is there, and D form of DNA, which is a rare form of DNA having only 8 pairs per turn. Another form of DNA is Z-DNA. This is left hand helix DNA. So we know BDNA is the commonly and naturally found in most living systems, that is right hand helix DNA, whereas Z-DNA is left handed helix DNA. This Z-DNA sugar and phosphate linkages follows a zigzag pattern. In this image, it is very clearly showing the turn and helix of the Z-DNA. How DNA, this is the secondary structure of that Z-DNA, and this Z-DNA plays a role in regulation of gene activity also. So the arrow marks are showing here the direction of helix. 4 types of DNA are there: A, B, C and Z, where A is the common and naturally found in most living systems. Okay, next one, functions and types of RNA. Very important. As we know, 4 types of DNA are there: A, B, C and Z forms. Here in the RNA also having 3 types: ribosomal RNA, messenger RNA and transfer RNA. Ribosomal RNA, messenger RNA and transfer RNA. Okay. Messenger RNA, let us see. For the first one is messenger RNA. It constitutes about 5% to 10% of total cellular RNA. It is a single stranded base complementary copy of one of the DNA strands of a gene. That it provides information of amino acid sequence of the polypeptide specified by the gene. See, these all eukaryotic microRNAs, messenger RNAs are monocistronic RNAs, that is coding for single polypeptide specified by single cistrons. Okay, and then ribosomal RNA, denoted as rRNA, occurs in association with proteins and is organized into special bodies of about 20 angstroms in diameter. Okay, it constitutes 80% of total cellular RNA, whereas messenger RNA having 5 to 10% of total cellular RNA, and ribosomal RNA is having 80% of ribosomal RNA and 5 to 10% of messenger RNA. Among total cellular RNA. Okay, major part is ribosomal RNA, and third one is transfer RNA. Okay, transfer RNA is also known as soluble RNA, nothing but sRNA. It constitutes about 10 to 15% of total cellular RNA. It is a class of RNA which is of small size of 3S type and generally having 70 to 80 nucleotides. So 2 types of nucleotides are there. 3S type and also 3S type is there, generally having 70 to 80 nucleotides, whereas the longest tRNA having 87 nucleotides. Okay, this S is nothing but Svedberg units. Okay, here in this slide it is very clearly representing the structures of messenger RNA, tRNA and rRNA. Messenger RNA is a single stranded. It is present in the nucleus and cytoplasm region, whereas transfer RNA is only cytoplasm region. The location of function in eukaryotic cells is given here. The messenger RNA carries genetic code for proteins, whereas the transfer RNA carries helps in incorporate amino acids into polypeptide chain, and ribosomal RNA, which constitutes 80% of total cellular RNA. Okay, the major function of ribosomal RNA is structural and functional components of ribosome. The locations are given here. Ribosomal RNA is present in cytoplasm, messenger RNA is present in nucleus and cytoplasm, and transfer RNA is present in cytoplasm region. Okay, these are the functions and types of RNA: ribosomal, messenger and tRNA. Now coming to the timeline of DNA and RNA discovery. Here it starts from 1833 to 1956. In the timeline, in 1869 majorly, if you observe here, in 1869 Frederick Miescher was the first person who separated cell nuclei from the cytoplasm and extracted an acidic material, nothing but nuclein. I mean, he called that acidic material as nuclein. Okay. I mean, and then after later on Richard Altman in 1989, he used the term nucleic acid, whereas Richard Altman, he used the term nucleic acid. And then after 1940, if you see here the timeline of DNA, the 1940 is one of the important one. Why? Because Erwin Chargaff, he worked on the amounts and ratios of different bases like purines and pyrimidines. I mean, Chargaff states that number of purine bases like A and G is equal to the number of pyrimidine bases C and T. I mean that if we came to know that A plus G is equal to C plus T, I mean Chargaff states that number of purine bases is equal to the number of pyrimidine bases. I mean A plus G is equal to C plus T. Okay. So and then after by the early 1950, X-ray studies of DNA by Wilkins, Franklin. I mean it shows the diameters, multiple. I mean it shows the diameters, and also in 1953, Watson and Crick they proposed a double helix structure of DNA, which quickly gained wide acceptance. That is a major breakthrough in the timeline of DNA. Okay. Majorly 1869, Frederick Miescher discovers the nucleon in the nuclei of white blood cells, and then after Griffith demonstrates the transforming principle in 1928, and in 1948 Chargaff and colleagues discovered the regularity in base of ratios of DNA and RNA, and also in 1953 Watson and Crick devised the secondary structure of DNA. Okay, and also if you look into the DNA timeline, major thing is Watson and Crick. The major scientists are Watson and Crick. Here is the image of Watson and Crick in this slide, and also the RNA. Frederick Miescher in 1868, who called the material nucleon, at that time only he started working on RNA. Okay, I mean we came to know that 1868-69 is the era where DNA and RNA discovery was started with the help of Frederick Miescher experiments. He found that material called nucleon. Okay, this is about the timeline of DNA and RNA discovery. Let us go to the next slide. Central dogma, the very important topic, very important thing here. We have to learn about the central dogma. Here, the flow of information, the flow of genetic information are of two types. One is the perpetuation of genetic information from generation to generation, and second one is the control of the phenotype, nothing but gene expression. Okay, in 1958, Crick proposed the information present in the DNA. What he proposed, the information present in the DNA is transferred to RNA, and then from RNA it is transferred to proteins in the form of amino acid sequence. So listen carefully. Crick proposed that information present in DNA is transferred to RNA. DNA is transferred to RNA, and then from RNA it is transferred to proteins in the form of amino acid sequences. And this information does not flow in the reverse direction, that is from protein to RNA, RNA to DNA. It is, I mean, this information never flows in reverse direction. In 1958, Crick proposed that information present in the DNA is transferred to RNA, and then from RNA it is transferred to proteins in the form of amino acid sequence. Okay, this relationship between DNA, RNA and protein is known as central dogma. Central dogma is nothing but the relationship between DNA, RNA and protein. So DNA molecules, however, provide the information for their own replication. DNA express its genetic instructions by first transferring its information to an RNA molecule by a process called transcription, and then after the transcription, the mRNA underwent for translation to form a chain of amino acids, nothing but polypeptide chain. I mean, the RNA molecule then transfers the genetic information to a protein by specifying its amino acid sequence. This process is called as translation. Okay, so I mean the information must be translated from the language of nucleotides into the language of amino acids. Here the name itself, translation is a complex process where involving ribosomes, tRNAs and other molecules. But the information must be translated from the language of nucleotides, I mean mRNA having nucleotides, into the language of amino acids. Polypeptide is nothing but it's nothing but chain of amino acids. Okay, the flow of genetic information according to the central dogma of molecular biology is DNA to RNA through transcription, RNA to polypeptide chain through translation. Okay, we can now identify three major pathways of information flow in the cell. In replication, I mean the information process from DNA molecule to DNA molecule. So that is one pathway. Information flow in the, what are the informations like genetic informations flow in a cell in two major pathways, like one from DNA molecule to other DNA molecule, and another through transcription and translation. I mean information passes from DNA to RNA, and in translation information passes from RNA to protein. This concept of information flow was formalized by Francis Crick, and he called this as central dogma of molecular biology, and he called this as central dogma of molecular biology. I mean that central dogma states that genetic information passes from DNA to protein in one way information pathway. I mean DNA to RNA, RNA to protein. We now realize, however, the central dogma is an oversimplification. Here two flows are there. One is perpetuation of genetic information from generation to generation through replication, DNA replication. So that we will discuss in the next slide. I mean DNA replications, conservative, semi-conservative and dispersive methods of replications are there. And another one is that control of the phenotype, I mean the gene expression. That is one flow of genetic information. DNA underwent transcription to mRNA, I mean DNA to RNA through transcription, and RNA to polypeptide chain, I mean chain of amino acids by translation. Okay, this is about the central dogma of molecular biology. And then DNA replication. So majorly there are three possible modes of DNA replication. What do you mean by replication? The process by which a DNA molecule makes its identical copies. The process in which DNA molecule makes its identical copies is called as DNA replication. There are three methods of DNA replication: semi-conservative method, conservative method and dispersive method. Okay, in this slide we are going to discuss about DNA replication, the basics of DNA replication. What do you mean by DNA replication? The process by which a DNA molecule makes its identical copies is called as DNA replication. Here three methods are there: dispersive, conservative and semi-conservative method of DNA replication, where semi-conservative method of DNA replication is widely accepted method. So dispersive method, the whole DNA molecule, I mean the parental DNA molecule, would break into several pieces. Each fragment would replicate the old and new segments, would recombine randomly to yield the progeny of DNA molecule. It is there in the figure that the first generation of progeny, it is like a mixture of old and new segments. Okay, I mean each progeny molecule would have both old and new segments along its length. That is dispersive. And second one, conservative. According to conservative scheme, two newly synthesized strands, I mean two newly synthesized strands following the replication of a DNA molecule would associate to form one double helix, while the other two old strands would remain together as one double helix. I mean two DNA strands form, I mean two new DNA strands are there, two old strands are there. Two new strands form a structure and two old strands form a double helix. Okay, here in this conservative method, two newly synthesized strands following the replication of DNA molecule would associate to form one double helix, while the other two strands would remain together as one double helix. Okay, semi-conservative method is widely accepted. In this model of DNA replication, each newly synthesized strand of DNA would remain associated with one old strand against which it was synthesized. I mean one new strand and old strand. Thus each progeny DNA molecule would consist of one old and new synthesized strand. So there is an experiment which shows why the semi-conservative method of replication is widely accepted and how the replication procedure, and in detail to know about the replication procedure we need to know about the polymerases, ligases, what are involved in the what are involving in the DNA replication, and also the replication fork and origin and all. It's a big topic that we can go into the another chapter. Okay, so with this I want to conclude that in this video I covered the DNA and RNA and its nature, and along with that I also covered the structures of DNA and RNA, types of DNA and RNA, where the types A, B, C and Z, and also the diagrammatic representation of double helix model, and also the salient features. Okay, along with RNA, what are the types of RNA, mRNA, tRNA, rRNA, and their functions and their constituent percentages in the cell, and then after central dogma, and after the timeline of DNA and RNA, and about Watson and Crick, and also and finally what are the two pathways of central dogma, one through replication and one through transcription and translation process. So this is about the nucleic acids, DNA and RNA and all. Okay, so I took these are the image sources. Okay, please follow if you are having these soft copies. Thank you so much.